actually is very recognizable. And so when you're looking at a flagella motor, when you're looking at a lumen or lysosome, this region, or the ATP synthase, these are examples of it. It's like a water wheel, it turns. There's actually physical motion that's turning this rotor. When you get into the fine detail, you'll actually find that even this unit has got two pieces to it that they turn. There's actually specific amino acids. The protein will protonate it, and then as the next one comes along, they'll actually push it the next position and push it the next position to accept the next proton. And it actually is very much like, you know, those that you spin and want to see what's going on. It's very much like that. It's got a trigger switch on it that goes one way as it turns that rotor. And it's actually, in its design, this is one of the weird enzymes you don't need to know now, but it's really kind of cool. ATP to synthesize, when we look at the energy curve, it almost takes no energy for the enzyme. Once it binds the enzyme, it makes ATP. So it takes ADP and inorganic phosphate, and it's done. However, to get it back off of the enzyme, it takes a lot of energy. And that energy is where the protein's conformationally changing as it does a rotation. And it really has a knob that changes the shape and pushes it off at the last instance. So it actually binds 3 if you don't allow it to rotate. We'll make 3 ATP on it, but you never get them off if you can't do that conformational change. Yeah.
So on the other two, like the T and ABC, should we be able to like pretty much draw out what happens in it, or just knowing like?
So you know in these instances that it's multiple choice. But a multiple choice question might include some, if we dreamed up one, something like, gosh, if this is a P-type active transporter, right, for sodium potassium pump, what are the 3 domains, right? And then I named them. That would be similar to what we did today, right? If we were saying, which of these has 3 active domains, and then I listed all the types of active transporters, that could also be a way to ask it. Just twist, you see examples. I also might ask which of these is an example of a P-type transporter, and then list examples that you should know and recognize off of the ones I gave you. So anything like that would be fair game.
Okay.
Does that help in expectations?
Yeah, that's good.
And that would be similar for all of those. Okay. Well, let's leave that behind. Again, on this tail end, these are here for your enjoyment, and you're capable of doing the math. It's technically there for you to consider doing as part of the study. I won't make you do this math on the upcoming exam. Okay? So if you're freaked out about it, let it go. It's okay. All right. Cool. So when I look at the schedule, it says today is Friday, Monday, Wednesday. Wednesday's also the beginning of the exam. So keep that in mind. We'll have a brief application actually talking about caffeine next week on Wednesday as well, and how it affects your memory. Is it good or bad? What do you think? We'll talk it through. Okay.
It's gotta be good. People enjoy that shit.
If I were giving you a precursor, chocolate, which contains caffeine and theobromine, is also known as an aphrodisiac.
Ah,
Which
crap.
means it's going to do what?
There you go.
Enhance the experience, but ultimately make it you're talking about a good experience. Okay? Something you remember because it was significant. Okay?
I gotta go buy chocolate before I take my exam today.
Then you do it before you study.
Yeah.
And then go take it.
Otherwise, you'll just remember
Okay.
things like failing the exam.
Yeah,
as
you're
well.
right.
Last
I remember all the
chance to share,
questions.
but just a quick moment here. I love this scripture found in John. My sheep hear my voice, I know them. It's the Savior speaking, right? And they follow me. And I give unto them eternal life, and they shall never perish, neither shall any man pluck them out of my hand. promise you, the Savior knows you. If you're committed to following Him, He has the power to save. He is our Savior. He can. He can save all of us. He's got that power. That's a profound realization to just digest. I'm going to do my part. And I'm going to let Him figure out how to get me to where I can't even imagine I'm going to be able to go. Okay. Signals. Hearing the voice, hearing the signal sounded good. All right. Wow. A lot of signals there. Androgens, hormones, neurotransmitters, light, touch, pheromones. What do we do with it? Cell division, growth, differentiation, antibody production. We got a lot of things to cover. A receptor is the target of, and some downstream signaling cascades is the target of our upcoming chapter 12. A membrane-bound or soluble protein or protein complex which exerts a physiological effect after binding its natural ligand. There are lots of these. Nuclear receptors are located in the membrane on the nucleus side, okay? They have to be very hydrophobic to get there because they're penetrating, going all the way in. Many receptors on the outside of a cell, but if you're thinking about something that's a nuclear receptor, realize that would be affecting transcription down at its core. There are other external ones like G proteins that part of the cascade affects transcription, but it's not the actual directly affecting. There are 5 features of signal transduction. This is on the study guide. There is an exam question that's dealing with these 5 elements, okay? So in looking at them, why don't you talk through what you think you're reading there on the slide with your group for just a minute. I'll give you like 3 minutes and digest this, and then I'll pick a couple things to make sure you got it.
So specificity, meaning that, yes, but you could, some I think receptors bind a class of ligands.
There's some broader ones, sure.
Yeah.
As far
But
as
they
like
have,
the end result, like it's very specific to like
yes,
The receptor would be on the membrane.
yeah. So, binds one ligand.
Like the signal would have to be hydrophobic.
Makes
All
sense, right? You don't want everything getting amplification is when you have, that's why we have two cascades, right? Because
Like a
you will have
one-to-one thing.
one receptor bind, and then it'll set off other receptors so that they can like amplify that initial.
think of like white blood cells in your immune system, right?
Yeah.
One gets activated like estrogen, it just kickstarts like a flood of that, like multiplies.
Amplification, like a speaker.
Yeah.
Whoa, that was deep.
This is when enzymes activate enzymes. Well, what did you say? I'm sorry.
Okay,
Modularity.
top level word specificity. Yes, you expect your molecule that you're trying to signal to be specific to the outcome you're looking for, right? I don't want to be signaling something and having it go off topic. Desensitization, does that make sense to everybody? In fact, the signal could actually, in some instances, let's use a literal example. If I go outside and it's a bright sunny day and I can't see anymore because it's so bright, how come in a few minutes I can? I desensitize my receptors so that I'm not overwhelming it. Same thing happens with your smell sensation. It's like, don't you notice it stinks in here? Or we get used to it. Same reason some people are used to smell a dog everywhere in their house. It's like, you could probably clean this place. Integration, when two signals have opposite effects on it, but this is really talking about the fact that two different signals are affecting the outcome. It's taking its points from not just one. Modularity, we're dealing with that where certain proteins, we start with a signal and then it has other downstream pieces that it affects, but those other downstream pieces could be, that signal could be used in multiple ways. So when we talk about cyclic AMP that we'll talk about soon, which many of you know, think, oh, that's a secondary messenger that could be used in many instances, and I'll show you that, okay? Amplification, a signaling cascades should make some obvious sense to us. Yeah, go ahead.
Sorry, for the modularity, you're saying, so like a secondary, can be used later on for something.
Yeah, so part of the downstream signal, whether it's a cascade or not, there's similarities, right? The initial receptor works with high specificity, but the downstream molecule that's being produced after that may have multiple parts that we could use. Wow. This is really the topic that we're really covering. G protein receptors, tyrosine receptors. This will be the bulk that we spend our time on. Those two. We will cover very quickly a guanylyl cyclase, ion-gated channels, in adhesion with integrin, which we technically covered almost all that I'm interested in covering there, and also that we can affect transcription. So we have lots of other things, and you'll see that, but these will be the two we talk about most, okay? So we start initially with the GPCR. Now, if you have never heard the phrase G proteins and GPCR, you've got a lot of catch up to do at this point, but it's not because you did something wrong. There's just a lot of information that's going to come at you really quick. And for those of you who already heard it, you're probably going to learn a whole bunch of new things you didn't know about GPCRs, okay? The G protein itself is a trimer. It's made up of what three subunits? Alpha, beta, and gamma, okay? When I activate it, what's the first thing that happens? Yeah.
You're liking that line, doesn't it?
Okay, so I've got a GPCR that's affecting the G protein. And once that's activated, I get a conformational change in what's actually happening at that moment. Yeah, it has to do with GTP, right? And is GTP being cleaved then, or is it being bound then?
It's phosphorylating. Right?
See, we're gonna have to know, right?
Yeah.
So
Fine.
these are the important things to pick up, okay? So here's a lot of GPCRs. The one thing you'd notice is integral membrane proteins. They all have classically been identified as having 7 transmembrane regions of helical structure that's going through them. So whatever was working with them, that's historically been inherited and used in lots of different GPCRs. The beta adrenergic receptor is the one that's talked about in great detail. If I am saying beta adrenergic, we are saying what is the signal? What's the molecule that activates a beta adrenergic receptor? Do you know? If I have to talk about a beta blocker, what are we talking about?
Is that norepinephrine?
Yeah, that's.
Yeah, norepinephrine, epinephrine. Yep, and epinephrine is really what we're talking about. The adrenaline response, that fight or flight, that's what we're talking about right there, okay? Here's your G protein and some fun nuts and bolts about it. G protein comes in here, and it turns out that the G protein is in its active form when it's got GTP. It's in its inactive form when it's GDP. the first thing that happens when the GPCR kicks it, it's still got GDP in there. It will push the GDP out and bind the GTP. Once that happens and it binds GTP, guanine triphosphate, right? Just like ATP, it actually is on a timer switch. And it will actually hydrolyze it back to GDP. It's a timer so much that it will turn itself off. That G protein is capable of turning itself off. That's cool. We have other enzymes that will speed that up, but this is an example of RAS protein. RAS is actually really involved in cell proliferation and cell division. It's really important. Turns out 25% of cancers have the mutation in RAS. This is the one that turns off the signal, and it doesn't do it. So it's got a signal, it's supposed to turn off that G protein, and it's not doing it, which means that I keep trying to divide my cells, and rapidly. So, does that make sense? With the mutation, and this is the scissor position, okay? If I use a GAP, a GTPase activator protein, what is it doing? It's increasing the rate that this switch will turn off. It does that by actually stretching what's attached to the G protein with the GTP. It stretches that adenine triphosphate, it stretches one of the phosphates and exposes that phosphoester backbone. So it's really susceptible to hydrolysis right there. And so that's really the goal. A GAF, a guanylyl nucleotide exchange factor, is, if you could remember that acronym, you'd know it for sure. It's exchanging. It's kicking out the GDP so it can bind the GTP. So that's what we're talking about. And so this would increase the replacement rate. This one increases the on-off switch time. Okay?
So one speeds, like, gives you more and one turns you off faster? Does that make sense?
Yeah, so again, if you're talking about exchanging, it means that I'm more responsive to the signal faster that I just got, okay? The other one says, don't keep the trigger on as long. I want a short burst of my signal, okay?
Okay.
All right. So there's always this alpha, beta, gamma subunit, okay? The one that's popping off that's carrying the GTP, I should also clarify, is the alpha subunit that's walking away, okay? All right. So, fight or flight, for those who are strictly chemistry, never did any biology classes, would be this idea that I have adrenal glands that are sitting right on top of my kidneys, which means they have access to the blood that's being filtered at a very quick rate through my body. If I could just release the signal there, it would move quickly through the body. That makes perfect sense. What is it going to signal? It is saying, I see a bear. I'm gonna have to run. I just have to outrun my buddy, not you know, I don't have to outrun the bear. But what's it going to release as it releases this intense energy for us is it's going to break down your glycogen storage. That makes perfect sense. Is it also going to affect the rest of it? Yes, it's actually going to break down lipids. It's going to access all your energy stores so you have that energy. It's going to increase your heart rate. This makes some sense. So all these things are happening, but the fastest fuel I can get is my sugar storage, right? So I'm going to get to those. All right. We need to know these words. And the ligand is easy, but if I say it's now an agonist or antagonist, sometimes those vocabulary words get a little confusing. Please clarify to your neighbor how you're going to keep them straight or how you do.
An agonist is something that like helps with the response. So if you want a certain response, you can give an agonist and it will give you that said response. If you don't want a response, you can give an antagonist.
Agonist.
Kind of like blocks the response. You wouldn't see it. It has to do with the receptors. Yeah, so it's like you're fighting against what you want.
Shows the signal,
But it has to do with
acts
the
the same way
receptors.
the signal works. An antagonist will bind, but like not let the signal.
Pretty easy.
Okay,
So an agonist is a
the
promoter.
anti-portion, again, I'm not doing what I was supposed to do. I'm gonna block that
Okay,
instead.
but- Thank you.
The agonist, I'm gonna do just what I normally would have done, it's just a molecule that will substitute for the natural ligand. All right, fantastic. This is all in green, all 7 steps you need to know. So if you were memorizing these 7 steps, the key here is to say, I recognize a normal GPCR response. Okay, and we're dealing with this with beta-adrenergic receptor. This should make a lot of sense logically, and you're on a multiple choice, so you're pretty good. Incoming signal, I've got epinephrine. It just signals as it binds to the GPCR with the 7 transmembranes, it does a conformational change. That conformational change is going to affect the G protein. The G protein with its trimeric form kicks off the GDP and binds GTP on the alpha subunit. When it's binding the GTP, the alpha subunit is released, which then goes over to another location. The one in this that's being identified is known as the adenocyclase. That is the enzyme that's responsible to make cyclic AMP. So it's a normal adenosine ATP that becomes a cyclical structure. We'll look at that in just a second. That CAMP actually then activates the PKA, which is a kinase. So it's a phosphorokinase A, meaning one of the first ones that we discovered. And kinases, in this instance, go and phosphorylate a plethora of downstream targets that are saying, break down my glycogen, break down my lipids, increase your heart rate. Start doing whatever it takes so I can escape the bear. And so it triggers all of that. This is the step that most people forget. That was step 6. We get there no problem. But if I keep this going and PKA is active and it stays that way, I'm going to have a problem. So in order to shut PKA down again, I actually need to stop this cyclic AMP. When we look at the structure, you'll understand that it's actually releasing the enzyme's ability to do its job, because without it, it will be tethered. So I've got to break the cyclic AMP. So this is a phosphodiesterase. A cyclic nucleotide phosphodiesterase goes and chops up CAMP. Now it's just AMP.
Is there a separate cascade that gets the cyclic nucleotide phosphodiesterase?
So the actual PKA turns on
the phosphodiesterase.
at the same time it's saying go do everything else. It's saying stop the signal that just created me.
So you have like a feedback loop.
Yes, you do.
Okay.
As soon as I have adequate amount of phosphodiesterase that will be produced and it's on, it will shut it down.
Cool.
Okay. That seems reasonable. Great. Synthesis of CAMP. You don't need to go through the mechanism, but we're just kicking off a pyrophosphate, making it look like CAMP. We are 3', 5' connected with them on the monophosphate. That's what CAMP is. This recognition should be something you could recognize that molecule. Remember that on the upcoming exam and final, there'll be many structures. You'd want to know that. CAMP is considered to be a secondary messenger. There are other secondary messengers we're going to want to know, but CAMP is the one that's popular. Okay, so once it's produced, here is some awesome stuff in the mechanism. I am not making you go through this, but this is showing you how the aspartate here comes in with its carboxylate, affects the water, right, making a hydroxyl group, which then has the ability to affect right over here on the 3' position of the adenosine, which then actually then attacks here and kicks off our pyrophosphate. So there's the mechanism, ASO 2020 in its gory detail, okay? All right. Thousands of glycogens will be released from that camp after PKA affected. This is an interesting little plot. One epinephrine molecule turns around and releases the alpha subunit, turns on the cyclic AMP, and this activates now PKA. Did you notice that I got here more than one of the alpha subunits that came on?
So I was wondering about that. Are those like other inactive alpha subunits that can be activated by the same
No.
beta gamma?
So once the signal is on with the GPCR,
Uh-huh.
available G proteins can dock and be started.
To make sure I understand what's happening in the membrane, they're all in the same raft, right?
Approximately, yes.
Okay, but they can move.
Uh-huh.
So you have multiple G proteins that can be reacted by the same
Yeah.
GPCR.
Yeah.
Okay.
So in this next one, you notice that I am making multiple cyclic AMP. I turned on an enzyme, it's not turned off yet, it just keeps doing its stuff with the adenyl cyclase. That's fine. I then have the ability to take of those and affect the PKA, turning it on. I got more than one adenyl cyclase to do that with. And then I've got the kinase here that's turning around and breaking down my glycogen. That's a phosphorylase. We get lots of glycogen breaking into glucose 1-phosphate, which then is transferred over to make glucose 6-phosphate, which goes through glycolysis and we get ATP at the end. If I am doing this in the liver, I'm going to release all that into the bloodstream. And in order to cross the membrane, I'll actually rip the phosphate off entirely. But if I'm doing it inside of muscle tissue, I'll just use it for direct energy, which means I actually don't need to use hexokinase as the first step in glycolysis at all when I break down glycogen. Well, that sounds really cool. So I myself go through all the math on that because my son was given an EpiPen because he was allergic to things. They didn't want to have him have an anaphylactic shock. So I look at how much actual adrenaline is in there. How much epinephrine's in there? Quantitate it, look it up, check it out. Figure out how many moles it is, right? That I'm injecting. And then I go through this checklist. And I'm going to tell you, this is so far off. You'd be getting like 25 grams of sugar out of it. I mean, nothing, like a candy bar. You'd be like, oh, you wouldn't be running and buzzed up all going crazy. It does a lot more than what they're showing you. I'm just telling you. It does a lot more. But the point is, the signal is amplified many, many times over. So don't worry about the counts. Just know that it's amplified.
So the circles with the green triangles, what does that signify exactly?
So they start using here and continues on for a long time. A red X means it's stopping that pathway. A green triangle means that pathway is being turned on.
Okay, so missing one in the activating of the PKA.
Yeah, so cyclic AMP, in order to turn this on, is going to release it. We'll see that in just a second. But it's definitely turned on, but it's not a change of the enzyme. Okay?
So it's not like an activation of an enzyme.
It's an activation. It's actually taking off the brakes. It's handcuffed, and you'll see it actually looks like it's handcuffed. And the cyclic AMP will release the cuffs. Okay? Glucagon, remember, is the signal that says I'm starving. Insulin's the signal that says I got lots of sugar. Got it? Makes sense. We already talked about insulin response too. Epinephrine comes in at this point. Makes sense. These are my feedback loops. You don't need to know all this awesome stuff. Cool. No, this is actually totally from another chapter. You don't need to know all that. Cool. I use it in another course. So I did redrive. Yeah. So remember, the G protein can self-inactivate, right? That's awesome. So if I have this signaling going on, what does a gap do? It means I'm going to turn off my switch faster, shorter burst. Okay, that was the gap. Okay? So let's see. The other part that I need you to sort of understand here. Yep, awesome, cool. We call it BAR. Stands for beta arrestin. If I said that you were arrested, what would I be saying?
Sorry.
You're detained. Good. Doesn't mean you're guilty of anything. We just detained you, right? Threw you in the handcuffs in the cell and detained you. Well, the beta arrestin is actually going to do the same thing. It's going to detain and put it in a cell for a minute. Okay, so what's going on is I've got this awesome tyrosine. I'm going to phosphorylate as part of desensitization that I talked about before, too much light. I'm going to come around and phosphorylate this, which will actually recruit BARC, which is a beta arrestin kinase. And when I've adequately phosphorylated this beta adrenergic receptor, it will recruit the BAR, which will actually pull this guy off the membrane. Do endocytosis, pull it off the membrane, make a vesicle. What would the fact that I just took this one who had a signal with epinephrine, it's now got a feedback loop, BARC has affected it by phosphorylating it, now beta arrestin's pulled it off the membrane and it's sitting inside of the cell. What would that do to my signal?
The number of signals you can get.
Yeah, it's not that I got rid of the epinephrine.
You're just not caring about it.
I just removed the receptor from the membrane, so for a moment I can't see it. And then eventually, what do? We pull it back to the surface and sample again. Is it still there? This is the way I calm down, so to speak. So beta arrestin allows me to desensitize in one aspect, or also just resample the surface. So you might guess, and you'd be right, that BARC is activated by a whole bunch of DKA. Yep. Makes sense. Okay. Yes, here's all that awesomeness showing you as well what happens when the epinephrine's bound into the GPCR. It's moving those 7 transmembranes. It's actually changing the shape of it, which is changing the conformational shape on the G protein, which is then causing it to kick off and bind GTP, and that's how it is placed. Okay. About 1,000 GPCRs in your genome, and we do not know the structure of all of them. The first one that we solved was not beta adrenergic receptor. The first one we solved was actually rhodopsin. And the guy that I worked with, that was his structure that he solved in all of its active forms, including dark activated, light activated. And when they do the crystal structure on them, the crystals change color depending on which form of the activation it's in. And so if you shine light on the crystal, it will also move the proteins and change the shape of the actual molecules and crystal. It's weird, okay? All right. 500 were for taste and smell, by the way. Here's 150, we don't even know what they are. Yeah, they're there. Okay. Camp itself is a secondary messenger. Does it look like it's doing a few things?
Yes.
So when we talk about that interchangeable signal, it can affect a lot of downstream targets, more than one. Is that the only list? No, turns out there's more. So camp, yeah, go ahead. Right?
What are these slides or ones?
Yeah, there's just lots of information and you're seeing lots of green, right? Yeah, so the take home from this and this right here is that as a secondary messenger, camp affects a lot of downstream targets, period. That's actually all you need to know. But we will talk more about camp, but that's it. And when we were talking about this, you don't need to know anything. Here, the reason it's under green, you do need to know what a bark is and what a bar is. The beta-adrenergic receptor kinase, the beta-adrenergic receptor arrestin. So. All right. So let's get into camp a little bit more. This is a scaffolding protein. Scaffolding proteins often exist inside of lipid rafts. What is a lipid raft?
Thicker parts.
Thicker, like instead of 30, more like 50, and it's got more cholesterol and also some proteins that are integral membrane proteins that are associated with that are put in there, right? Okay, so it's more rigid. So a scaffolding protein is connected to the membrane, and you can see it's got anchors that are involved in it. It recruits different, usually enzymes, to sit right next to each other. In this case, there's our pKa. And pKa has these right here on the outside of them. So these catalytic subunits right here, which look like the handcuffs themselves, are actually ready here for the regulatory subunit to block it. But if I get into the regulatory subunit 2 units of CAMP, the handcuff comes off, the catalytic unit of pKa is now active, and it starts doing its job. And it goes after a whole bunch of downstream targets. But if I shut down my CAMP concentration, cut it up, making it AMP, then that will turn off. I used what to shut it off? What did I cut up CAMP with?
Yeah.
A phosphodiester.
Phosphodiester.
Okay. Well, that's pretty cool.
So
Ask.
the subunits, the handcuffs, just because you cut off cyclic AMP, are those going to immediately come back? Are they still like a part of the protein? Do they float away?
So they will rebind with the regulatory subunit. There's a high affinity for that. They like to connect and restrain CAMP. Okay. And so, but as long as they have adequate cyclic AMP, they're going to start working.
So is it like that there's like a level of cyclic AMP that's there?
A concentration.
And as long as you have a certain concentration, you're active. If you would go below that concentration, then you deactivate.
Yeah.
Okay.
Okay. Sounds reasonable. Yes, here's all the active catalytic site of cyclic AMP, which you might notice is affecting, here's an ATP, substrate comes in, it's got some hydroxyl group, and it's phosphorylating. It's a kinase, right? So pKa, a kinase, and it goes after not one target, but many, many targets. So that's what this little blue cuff is really doing in a little bit more detail. that you don't need to know. Okay? We just talked all about beta-adrenergic receptor characterizing GPCR. We're going to talk about some other GPCRs. Well, at first, when we come in here, this hormone-specific receptor receives its message, a hormone comes in, and it releases the alpha subunit. In this case, instead of affecting adenocyclase, what does it do?
It's activating the
Phospholipase C. So, okay, a phospholipase is responsible as a lipase for
the C part.
cleaving up fats, a phospholipase itself. So that's its job, and you can see here it's affecting what we call PIP2, which is this right here, this whole molecule. And what am I going to do? I'm going to cut it up so that I can make it into the inositol triphosphate, 3 phosphates right here. So essentially, phospholipase C that you saw before is now cutting off this molecule, which becomes a secondary messenger. So IP3 is a secondary messenger, which docks here to an ion channel, opening the ion channel, releasing calcium to come out, which then affects back at the cell the protein kinase C. That's a kinase, so it's going to phosphorylate a whole bunch of, hydroxyl terms, which then affect downstream targets, so, of the hormone. So there's a lot of inner circuit. So calcium is considered a secondary messenger. IP3 or inositol triphosphate is considered a secondary messenger in addition to KIM. So right now you have at least 3 secondary messengers in your working vocabulary. If I ask you a question, which of the following is or is not, then you could identify that.
So just as a kind of an application question here, with certain medications, like there's a drug class of calcium channel blockers. Is it this kind of channel we're looking at to affect blood pressure?
It is.
Yeah. So,
Yep.
okay.
You're right, Kevin. Yeah.
Well, I just wonder, what is it that makes it or defines it as a secondary messenger? Is it just?
The message that it is being carried from the active receptor or downstream created by it, but it's not the initial message, but it's a message instead of an enzyme. Okay? It's something itself. Yeah, go ahead.
Did you want us to know the pathway shown or just that?
No, thank you for asking. You do not, the only one I want you to know is the beta-adrenergic receptor, those 7 steps, and also how I turn it off or desensitize it. So this pathway, do not freak out. I only want to show you the other one. Go ahead.
Isn't isobutanol considered a secondary message?
Yeah, it is. Yep, it is. It's also a secondary messenger that's being affected here. But, all right. Calcium itself, there's a lot of things that can bind to calcium. Okay, calmodulin right here is the one that we're going to talk about that are being affected. Calmodulin kinase activates crosstalk pathways with cyclic AMP. It is not essential you know any of this at all. What I'm trying to teach you is a molecule by the name of calmodulin is specific enough to recognize calcium as a secondary messenger and can actually trigger based on concentration. If I were looking at it, talk about things like my muscle contractions, talk about things like heart rate, it can trigger these pulses. And yeah, tenfold difference in concentration is sensitive to it. And usually that's by opening and closing the calcium channel that I'm doing that. So whenever we see these channels opened right here, we're gonna close it milliseconds later. And I'll show you some that we talk about that timing later on in this unit, in this chapter, but milliseconds. They're not left open very long. Okay. The next one that we will talk about I'll start now because we have time, but it's really the focus for Monday, is the tyrosine kinases, okay? So before I go into tyrosine kinases, ask me any other questions you want on the TPCRs, because several good questions have been asked. What are the expectations? This chapter is huge. The study guide question on it looks long and daunting, but when I pull it up, let's see if we sort of focus on our chapter 12. For the following sections, know the general idea, not a detailed mechanism unless you like it. That will be actually dealing with some pathway receptors for different things like sight and smell, okay? General features of signal transduction, that was the 5 that we looked at right at the very beginning. Describe the initiation, propagation, and termination of a G protein signaling cascade. How is protein kinase A activated? Signal amplification is seen in the epinephrine cascade. Got it. How does cyclic AMP activate enzymes? Adds a phosphoryl group to it, right? Describe the signaling pathway illustrated under this, which is really what we've already got. And then calcium is a secondary messenger. This is an interesting question. How do alpha proteins interact with similar receptors and always act on the same downstream enzyme to create a secondary messenger?
Wait, okay.
Yeah. So they're always acting on the same downstream enzyme to create the secondary messenger, but the signal that came initially is not always the same. So how do they do that? Scaffold.
Or the bounded scaffold.
Got it. Okay. And then we'll come back on calcium, right?
Extra credit.
So, 5 minutes, let's use our time wisely. Now you want to pack up, but there's so much here. You might as well use your other 5 minutes. Tyrosine kinase. If it's a kinase, what do you think it's phosphorylating? Tyrosine. That's its target. So the protein kinase C, PKA, they were phosphorylated. But turns out that the one that we will look at is insulin as our model system. It's a tyrosine kinase. There are more. So I'm going to show you another slide in 2 seconds that has a whole bunch of other ones. This is the one we care about. It's a dimer. It exists in the membrane as a dimeric form. Incoming insulin binds to it, activates it, and it tends to then phosphorylate itself in its now active form. That's interesting. And so it's got 3 tyrosines on each of these dimers, changes its shape, exposing those to be phosphorylated, and then phosphorylates downstream things. There's a whole bunch of other tyrosine kinases. You do not need to know all those. Insulin's the only one we're looking at, and you just care about the big signal of insulin, which says, I have lots of sugar, I don't want to go through non-enzymatic glycation, right? All right, so much cool stuff. You've already seen this picture before. This was talking about GLUT4 transporter, insulin coming in a muscle cell, saying bring the GLUT receptors to the surface. So that little green arrow we talked about now is essentially saying, I'm going to phosphorylate so that I can change the solubility of this and push these receptors to the surface. Okay? All right. So they're being stored in vesicles, but they're being activated. ERK, you're not going to need to know ERK, ELK, and whatever, okay? But these will be where I'll walk you through it and we'll come back and we'll do it again, okay? You don't need to memorize all this. But the insulin signaling cascade is really interesting. Autophosphorylate itself, now phosphorylate IRS1. You always wanted to get the IRS, didn't you? This is your chance to get the IRS. And then it has 3 phosphoryl sites. It actually goes to a GERP and SOS. This is RAS. We just looked at RAS in this discussion today. What was RAS about? Yeah, RAS is a so RAS up here, the G protein. So its tag here is connecting it to the membrane. So RAS itself has a GTP, affects downstream, MEK, phosphorylates, ERK, these are kinases. They've all got this K in there because they're kinases. You don't need to know all their names. But the downstream signal is affecting what?
Our nucleus.
It's affecting the nucleus. Tell me about if I had sugar in the last 1 minute that I just am pulling in, what would I want to be affecting at the transcriptional level of my DNA if I'm now pulling in a whole bunch of sugar? I want to upregulate metabolism. Good job, Caleb. So you don't know all the enzymes, but you're like, I'm going to do glycolysis. I better do glycolysis. I need all the enzymes to do that.
Isn't the first step you need to phosphorylate the glucoses so they don't get out of your cells?
Yep, so I'm going to do that with hexokinase. That'll be our goal. Now, downstream, I'm also going to want to affect cellular division. Because if I've got a lot of sugar that's time to divide, I'm probably going to want to make all my ATP, but after I've made ATP, depending on the cell type I am, I'm going to store some as glycogen, and you will, okay? All of these are downstream targets. I'm also going to want to turn off, because I'm pulling in sugar, making things like glycogen. Don't make glycogen if I'm pulling sugar in like left and right. But stop breaking down fat. I'm burning sugar right now. So there are things we turn on and off. So that will be part of our discussion as we get into this, and we'll talk about even the crosstalk exists, like we talked about in that integration. Okay. Have a great weekend, you guys. It might snow some more, they say. Probably not.